Hello, everybody. Welcome to the Biology 142 recording for the snow day of January 25th. So sorry we missed this day. Would have been fun to do the lab. Please read your class messages from the class shell. I put forward a new schedule changes. So we'll be seeing each other next Monday. And please look at these schedule changes, and we'll start working on making up this lost time. And part of making up this lost time is this recording of the lecture material. So we'll be finishing up chapter 16 and starting on chapter 17. And what I'm going to do is go to this slides screen share. I will also have an animation. I hope everything will start fine. So let's get started. And before I do that, let's see if there's a screen sharing function that's working. This is exactly what we needed. So here we are in chapter 16, where the overarching theme is the consciously detectable, controllable activities of our nervous system. And here for a few minutes, we'll be talking about the activities of our nervous system that play a part in conscious control. While the only target organs we consciously control in our body are skeletal muscles. Way back in chapter 10, you have seen how nerve impulses were necessary to start a contraction of a skeletal muscle. And if you ever ask, where do all those nerve impulses come from? Well, you're looking at the answer, the primary motor cortex by the name of the gyrus. You know what? Let me see if I can just make this full screen. Right. So if you follow the cursor, the precentral gyrus is the name of this ridge in our brain. And when you look at these black lines, these represent clumps of neurons that start the nerve impulses that eventually will end up on skeletal muscles. So this is where all nerve impulses come from. That eventually guides skeletal muscle contraction. So it's probably comes to no surprise to you that there's not only a starter neuron involved here, but also other neurons in this passing down the nerve impulse to skeletal muscle. And so now that you know where all nerve impulses are coming from, we're going to take a look at how the nerve impulse starting from the precentral gyrus gets to those skeletal muscles. So these black lines, these clumps of neurons collectively are called upper motor neurons. And yes, you guessed it, we will be seeing lower motor neurons as well. So let's take a look at the rest of this nerve impulse conduction pathway. Again, the receiving end of these nerve impulses will be skeletal muscle. I'm going to skip through this slide. We will be looking at direct and the indirect pathways. So these are two separate ways nerve impulses can be channeled to skeletal muscles. And we're going to start with the direct one. Take a look at this slide, precise muscular movements. So when you are looking at your smartphone, find an interesting picture and you want to edit it, the guiding of your fingertip to that edit button is one of these precise muscular movements. So to make that happen. Nerve impulses are flowing through the direct motor pathways. And let's take a look at the actual pathway. Two neurons participate in this. The upper motor neurons start from the precentral gyrus. This black dot is the cell body. Take a look at this long axon, goes through the brainstem. And in the brainstem, in the medulla, most of the neurons cross from left to right. And of course, the neurons that start up on the right would cross over to the left. The rest of the neurons that's still part of this pathway will cross over in the spinal cord. So if you are pointing at that edit button on your phone with your right index finger, the left side of your brain, the left precentral gyrus, make that motion happen. So this is the simplest, quickest, fastest, most precise control of skeletal muscles. And when you look at this image and asking yourself, where are the skeletal muscles? You would have to follow these red lines all the way to muscles, and all 700 plus skeletal muscles receive nerve impulses in this direct pathway if you want to create a precisely controlled movement. So you see the role of the spinal cord here, and imagine if you want to move your arm, forearm, or if you want to extend your index finger to point at that edit button. Indeed, this is exactly where nerve impulses would go through. However, think about muscles that move the jaw, the mandible. In AMP1, we looked at several of these muscles. What about muscles that close the eye, close the mouth? Those muscles are so close to the head that, and so close to the precentral gyrus, that the precentral gyrus sends nerve impulses through cranial nerves. So these cortico-bulbar tracts are part of the direct pathways, but they involve cranial nerves. The others, they involve spinal nerves, as you see here. So let's take a brief look at just what are those muscles that receive nerve impulses from these cranial nerves that are listed here. It's too bad that we have to postpone that vision lab, because part of that lab would be naming cranial nerve 3, 4, and 6 as cranial nerves that deliver nerve impulses that move the eyeball. What about cranial nerve 5? This brings nerve impulses to muscles of mastication, masseter, temporalis muscles, just to citing the muscles from the AMP1 muscle lab. Cranial nerve 7 brings nerve impulses to muscles of facial expression. And if you want to look at Justin Bieber's viral infection that disabled his cranial nerve 7, you see that facial expressions can go haywire when there is no nervous control. In the AMP buzz section of your class shell, I think the very first post that I wrote, that is shared there, is about Justin Bieber's failure to maintain proper facial expressions. Injury to cranial nerve 7 does that. Cranial nerve 9 and 10 participate in swallowing, so movement of the skeletal muscles of the pharynx. Cranial nerve 11 is bringing nerve impulses to trapezius, sternocleidomastoid muscles. And cranial nerve 12, the last one here, brings nerve impulses to the tongue. So all these structures are located either on or near the head, and cranial nerves take care of bringing the nerve impulses. So there are no lower motor neurons for the cortical bulbar tracts, cranial nerves replace those. And here's the other way to get nerve impulses to skeletal muscles, the indirect pathway. So the reason why we have this separate pathway is because we have automated movements in the body. Automated movements such as maintaining the balance, maintaining erect posture, maintaining a particular set of automated movements such as walking, climbing stairs, riding a bicycle. These are all activities we don't have to pay close attention to. These are not high precision movements. They have been in our lives for years, sometimes decades, and they became automated. When we look at the automated movements, we do have the motor cortex that originates the precentral gyrus that originates all these nerve impulses. But the precentral gyrus creates a huge excess of nerve impulses, and we need editors, supervisors, if you will, to weed out these large number of nerve impulses and keep only the ones that we actually need for smooth skeletal muscle movement. So when you look at these boxes, basal ganglia, thalamus, cerebellum, motor centers in brainstem, such as substantia nigra, red nucleus. These are all names of the editors localized in different parts of the brain. This particular slide emphasizes the basal ganglia. These are the basal ganglia are clumps of neurons located at the medial aspect and the basal aspect of the cerebrum. The thalamus, we talked about this already, is part of the diencephalon, the robin egg-sized clump of neurons. The cerebellum and the brainstem are additional parts of the brain, again participating in this editing function. So whenever one of these editors are damaged, whether it's a physical damage or a malfunction of the neurons because of some genetic disorder, skeletal muscle movement becomes difficult to control. Jerky muscle movements, sometimes near impossible situations of difficulty moving muscles, such as described here as muscle rigidity, uncontrolled activity as tremors. So Parkinson's disease and Huntington's disease are two genetic disorders that disable basal ganglia and also some of those centers in the brainstem. Another organ, another part of the brain whose injury or malfunction leads to a malfunction of skeletal muscles in the automated movements is the cerebellum. So cerebellum is great because this is receiving nerve impulses representing the intent for movement, and this set of nerve impulses is coming from the precentral gyrus. The cerebellum, if you remember way back in chapter 16, the beginning of chapter 16, the cerebellum receives nerve impulses from proprioceptors. So those muscle spindles, tendon organs send many of their nerve impulses to the cerebellum. So the cerebellum actually knows what the body is doing in three-dimensional space. So it can monitor actual movement. The big achievement of the cerebellum that it compares the intent with the actual situation on the ground, and it can change the directions from a precentral gyrus. So the cerebellum is essential because of these abilities to maintain automated movements. The final common pathway to bring nerve impulses to skeletal muscles are nothing else but the lower motor neurons. These are, again, going through either spinal nerves or cranial nerves, and the rest of the structures are participating in the direct pathway. So the lower motor neurons are playing a role as the last neuron to take nerve impulse to the skeletal muscle in the indirect and in the direct pathway. And so that's all we're going to say about conscious control of skeletal muscles. We will be talking about skeletal muscles in AMP2 quite a bit. Maybe the first time skeletal muscles come into the picture will be the respiratory system, where inhalation, voice production depends on that, in the digestive system, mastication, activities in the oral cavity, movement of the tongue for swallowing will bring us yet again skeletal muscles. And then in the digestive system and in the urinary system, we'll see that conscious control of urination and defecation are also tasks of skeletal muscles. So we'll be seeing them. And again, in the lab, in the eyeball, and it's coming up in Monday's lecture as well, the skeletal muscles that move the eye. So we'll see references to skeletal muscles in future chapters as well. I think it's a good idea, even though we may have covered part of this in the previous lecture, to look at the integrating functions. And this includes wakefulness and sleep. A part of our brain, the hypothalamus, knows, based on visual cues, when we're supposed to be awake, when there's daylight, and when we're supposed to sleep, when there's no daylight. The hypothalamus sends out nerve impulses to control this middleman, the reticular activating system. And the result is either wakefulness, consciousness in our cerebrum, or sleep, which is an unconscious state of our cerebrum. So here's the middleman, the reticular activating system. Take a look at this little part here. Here's the hypothalamus. The hypothalamus sends out nerve impulses to the reticular activating system, which is located in the brainstem. And in the morning time, the reticular activating system will send out nerve impulses to the cerebrum, telling it to turn on, so we wake up. And at nighttime, the reticular activating system lowers its nerve impulse output, and the cerebrum is allowed to turn off to go into an unconscious state we call sleep. With the sleep portion of this chapter, will stay very, very short. It's good to know that the sleep is divided into several segments. These are alternating, starting with the non-REM and then the REM stages. Each of these lasts maybe an hour and a half or so. The restful sleep, the relaxation, the rejuvenating sleep is coming from this stage. We very poorly understand just why we do need this REM stage, but sleep research and many hospitals now have sleep labs to identify sleep disorders. Missing out on this portion of the sleep, the REM portion, can lead to sleep disorders and just a poor quality of life, because once somebody wakes up after a poor sleep, the whole day is shot. There's just little energy and motivation to do anything. There are several ideas out there just how sleep comes about. In addition to the nerve impulses from the reticular activating system, there are theories that chemicals must be involved in the brain. And this is what you see here, this idea that caffeine can prevent us from falling asleep because it counteracts the chemical adenosine to act in the brain. All I'm going to say is that we understand very little about all this, and true caffeine and many other chemicals can postpone the onset of sleep. Exactly how they act is still out for jury. The last aspect of our consciously working, consciously detectable, integrating part of our nervous system, which is the brain, is learning and memory. And you will be, hopefully you're already doing this, the learning part and building up a memory of what's the content of the class, because you will have to recall that memory, that stored knowledge at lecture exam 1 and practical 1 time. So this is essential for making our lives better in general, because once we solve a problem, once we face a situation and successfully navigate it, it's such a useful thing to store away that experience, that knowledge. So for learning, we have to have memory, we have to have storage of that previous experience and just how we dealt with it. So we have a short-term, immediate-term ability to retain information. The physical location, anatomical location for these neurons is in the hippocampus portion of the brain. Long-term memory is stored in the cerebrum and requires anatomical changes. Now, the anatomical changes, don't think that a new lobe of the brain needs to grow. Anatomical changes means that neurons develop more connections. More synapses will be appearing, branches to axons will appear and make connections with other neurons in a more abundant fashion. This takes a long time to develop because this requires actually building new cellular structures. But again, the anatomical changes are at the cellular level. So the cellular anatomy of the neurons develop more complicated connections. Both the sleep and wakefulness and the learning and memory are topics where we could spend a semester easily in each of those. So we are not doing justice for this portion of the nervous system, but this is our curriculum, so we're going to leave it at this. What I'm going to do is switch screen sharing to chapter 17, and let's take a look at the special sense of hearing and other mechanoreceptor related special senses. So give me a second and we will be starting on chapter 17. And let's see if I can get chapter 17 on screen. Oh, here we go. I might make this full screen again. All right. So with chapter 17, we are still in this consciously detectable, consciously working portion of our nervous system. Here, instead of having the receptors, the neurons scattered throughout the entire body, the receptors are concentrated, aggregated into a small area. In the first lab, you have seen the special sense of hearing. So I think I mentioned there that the cochlea that actually contains the hearing-related receptors have a size of a chocolate chip, and outside of those two cochleas, we have a left and right cochlea. Outside of those chocolate chips, there's no neurons in the body that could help us with hearing. So, as opposed to the warm receptors or touch receptors that are scattered through the skin, a 10-pound organ, now we have just two chocolate chips. And that's the drastic difference between the density of receptors and the concentration of receptors between general sense and now here in the special sense class. So let's go through these gross and microscopic anatomy, and we'll have an animation here that explains quite a bit, I think a lot more visual fashion than any models could in the lab. What you'll see is most of the information will overlap with the lab activity. The physiology part will be a little extra, and we'll have a little more information on the static and dynamic equilibrium as well. So when you look at this image, this looks very, very similar to the ear models in the lab. And we can start by just naming the regions here. So from the auricle to the eardrum, this is what we call the external ear region. You see a nice view of the middle ear. This is the second region. And here, again, the third region, the inner ear. We are seeing roughly the same picture on our lab model as well. So we'll be taking one region at a time. And let's start with the external ear. In terms of the detection of those pressure waves in the ear, the sound waves, the auricle and the external ear canal are just funneling and channeling these pressure waves to the last part of the external ear. This is the eardrum, and this is a thin but living tissue that starts to vibrate when those pressure waves hit the eardrum. So here's some of the contents that make up the eardrum. So we have connective tissue, and we have an epidermis-like covering. But the point is that this is living tissue with living cells. So when the eardrum is injured, whether it's some kind of an explosion, some kind of a trauma that ruptures the eardrum, or when during ear infection a tube is placed across the eardrum, those injuries to the eardrum start a healing response and scar tissue forms. A lot of scar tissue on the eardrum will lead to hearing loss. The eardrum needs to maintain its thin nature to be able to vibrate when those pressure waves hit it. Switching on to the middle ear, this is a lot more elaborate view than what we see on the model. And nonetheless, the very basic is that once the eardrum vibrates, we use 3 auditory ossicles to take this vibration all the way to the inner ear. So the middle ear is kind of a middleman, and you see the hammer glued to attach to the eardrum. Once the hammer vibrates together with the eardrum, the hammer makes the anvil vibrate, and a vibrating anvil will make the stirrup vibrate. So we are passing on pressure waves all the way to the doorstep of the inner ear. Interesting aspects here. is that the middle ear is connected to the nasopharynx through this auditory or eustachian tube. The auditory ossicles are suspended by ligaments. And we also have two tiny muscles here. Here's the remnant, a lot of it is cut away, the stapedius muscle. And here's another somewhat larger muscle, the tensor tympani. These skeletal muscles are programmed to contract and slow down the vibration of the auditory ossicles when a sudden loud burst of noise comes to the eardrum. A loud noise represents very, very strong pressure waves, and an intense vibration could damage these small bones. So we have this reflex that when we hear a loud noise, the two muscles contract and dampen the vibration of auditory ossicles. And let's see, let's go to the inner ear. The ancient Greek name for the inner ear is labyrinth. Labyrinth just means maze. When the inner ear was first dissected, the scientists saw that there were liquid filled canals, but that they also saw that these canals formed all kinds of different shapes and inlets and outlets. So, of course, in those days, in the 1700s, they had no idea what the inner ear, what the labyrinth was good for. So we have very ancient names like the cochlea, the vestibule, and the semicircular canals with the ampulla. So what you see on this image is the membranous labyrinth. The membranous labyrinth is holding this endolymph in all these colored portions that you see here. The bone layer around it is part of the temporal bone because the entire inner ear is encased in the temporal bone. I wanted to show you this aspect of the inner ear. In the lab model, we've seen cranial nerve 8. This is the cranial nerve that collects sensory nerve impulses from the cochlea. But on our lab model, it's not really clear that the cranial nerve 8 also collects nerve impulses from the ampulla. We'll be taking a closer exploration of the ampulla and also from the vestibule. So this is the first indication that in addition to receptors in the cochlea, we must have receptors in the vestibule and receptors in the ampulla as well, because nerve impulses are being collected by the cranial nerve 8. So this is a good artistic rendition showing the location of those hair cells in the ampulla and the vestibule, just by the virtue of the bundles of axons collecting nerve impulses from those places. When it comes to the physiology of hearing, we have to look at this static view. This is a good way to illustrate pressure waves hitting the eardrum, the subsequent vibration of the ossicles, malleus, incus, stapes. And then what the stapes, what the stirrup does, it taps this vibration into the liquid-filled portion of the cochlea, starting with the scala vestibuli. And then this pressure wave at the top of the snail moves over to the scala tympani, and the pressure waves rumbles out of the cochlea at this oval window. I'm sorry, it's a round window. So what the stirrup does is what your hand can do on the surface of a liquid, whether it's a lake or a bathtub. When you move your hand up and down on top of water, you create ripples. Those ripples on the water are pressure waves. So that's the best illustration that physical action in a liquid can create pressure waves. It happens here. The stirrup pumps these pressure waves into the scala vestibuli, which moves through the scala vestibuli and comes out of the cochlea to the scala tympani. This colored portion here is the cochlear duct. We'll be closing in on that to describe the action. This is very similar to one of our lab models showing the scala vestibuli up here, the scala tympani. Again, pressure waves are rushing through there. And the cochlear duct, which is filled with endolymph, has the organ of Corti with the sensory receptor hair cells. And this entire structure, you'll see this on the animation, will be moving up and down when pressure waves are moving through the scala vestibuli and tympani. It's also a good illustration how nerve impulses are collected from the organ of Corti, the cranial nerve 8 bundle reaches that point. It's good to look at this magnified view of the organ of Corti. These yellow structures are the hair cells, and you can see these hairs sticking out. When the entire organ of Corti and cochlear duct starts to jiggle, these hairs are pushed into the tectorial membrane, and that bending of the hair cells will be starting the producing nerve impulses. So let's take a look at the animation here, and then we'll come back to look at actual hair cells. We'll be coming back to this view to see what happens inside hair cells once the hairs on the top are bent. Now, just one more word about these hair cells. These hairs that you see here, these tiny yellow extensions, are not like hairs grown out from the skin. These are microscopic structures. These are barely visible under the microscope. The cells are cell size, so the hairs are extremely vulnerable extensions of the plasma membrane. It's just the naming hair cells that I guess comes from the skinny nature of these extensions. So let me see if I can go to a different share and share the animation. And I think we're going to go here. Let's see if we still have this right. Let's make this full screen. So what you are seeing here is the inner ear with the stirrup. And I'm going to stop this animation periodically. We're going to zoom in onto the organ of Corti. So let's start this. You'll be seeing the stirrup vibrating, pumping the pressure waves inside the inner ear. This is just the demo movement of the stirrup. We'll see a really high-speed animation in a few seconds. This is the low-speed vibration of the stirrup, and here's the high speed. So this is what our stirrup does when we are here listening to music, sounds, and all those pressure waves will be traveling into the cochlea. So here's highlighted in green the cochlea, and we'll be getting close to that. Here are pressure waves driving through the scala vestibuli and scala tympani. So let's take a look at here comes the pressure wave out through the scala tympani. And very soon in pink, the cochlear duct, the scala media, appears. And we'll be seeing just how this jiggling of the entire cochlear duct happens. So here's our close-up. I'm going to stop this. Take a look at the one good aspect of the animation, show the scala vestibulae up here. Highlighted in green is the vestibular membrane. Down here is the scala tympani. Pressure waves will be rushing through here like crazy. And in addition to the vestibular membrane in green, a little later, the basilar membrane will also appear green. You see the tectorial membrane, you see the organ of Corti, and once we see the jiggling of this entire cochlear duct, we'll be zooming in very high magnification on the hairs and the hair cells. So let's take a look at this jiggling first. So here's in green the basilar membrane, and here comes a pressure wave, and take a look at this jiggling action. The entire cochlear duct is moving up and down. And this is where the hairs need to be bent as the hairs are moved into the tectorial membrane. So we're going to close in, zoom in on that central part. As soon as the jiggling stops. Right. So I'm going to stop this right here. So in green, now you see the hair cells. The hairs themselves are these tiny little extensions. We'll have a little better look of that. The tectorial membrane is this giant overhang up here. It will be highlighted in green as well. And then this entire structure will get into motion. So let's see if I can start and catch this on time to show the essential function. So here in green is the tectorial membrane. And here comes the jiggling motion. And I stopped it right here. You can see the hair cell with the hairs. And this is how the hairs are bent. This up and down movement push the hairs against the tectorial membrane. And from a straight position, they go to a bent position. And this is where chemical changes happen in the hair cells. From lecture slides, we'll pick it up from here, but I wanted to show you that this is what happens every time we hear sounds. These hair cells in the organ of Corti and with the tectorial membrane moving up and down, jiggling, and in that jiggling process, the hairs on the hair cells pushed into the tectorial membrane, so they achieve this bent shape. So let me go back to the lecture slides. And we are going to screen share from here. So here is now the artist's rendition of these hair cells. You can see the hairs, these extensions. Once the hairs are bent, the first action that happens here is potassium channels will be opened. These are mechanically gated. You see the little lid here that's being moved out of the top of the channel. And so once these green potassium channels are open, potassium ions, which is highly enriched in the endolymph around the hair cells, potassium ions will rush in. So channel opens for the potassium ions, it rushes into the cell. This high potassium concentration inside the cell will open voltage-gated calcium channel. These calcium channels allow now calcium ions to rush into the cell. And calcium ions in the hair cells will allow neurotransmitters to be released. And let me see if I can move this bar around. Just for this. So underneath the hair cells, there is the beginning of cranial nerve 8 neurons. And when neurotransmitters are released, this blue neuron will now fire a nerve impulse. So this is how nerve impulses are created for the special sense of hearing. Again, after that long path of bringing pressure waves all the way into the cochlea, we achieve the bending of the hair cells in the organ of Corti, and then those hair cells release neurotransmitters that stimulate nerve impulses from the neurons of cranial nerve 8. Of course, this nerve impulse needs to go from the cochlea all the way to the temporal lobe. So cranial nerve 8 is taking nerve impulses through the medulla oblongata. Nerve impulses will go through the thalamus portion of our brain, and the target, the destination, is the auditory area of the temporal lobe of the cerebrum. We did some experiments, the Rini and Weber test, that can identify conduction and nerve deafness. So plainly stated, conduction deafness is related to damage to either to the eardrum or the auditory ossicles. Nerve deafness is related to the structures that participate in action potential nerve impulse production, the hair cells, and neurons of the cranial nerve 8 itself. With the equilibrium special senses, we mentioned these in the lab, both the static and dynamic equilibrium. You have seen in the lab at least describe the macula associated with hair cells of the static equilibrium. We had in the lab also mentioned crista as a location of hair cells for dynamic equilibrium. So let's take a little look at how those hair cells are located in a way that they do not react to sound-related pressure waves. And the answer is, take a look at, we are in a vestibule, so we are looking at a set of hair cells inside the, or part of the macula. This is also called the otolithic membrane. Take a look at these blue hair cells. They are encased in a jello-like material, so sound-related pressure waves cannot move these hair cells. What moves these hairs is the following: bending of our head, forward, backward, or sideways, will displace this jello in the otolithic membrane, and this movement of the jello layer is going to bend the hairs. Once the hairs are bent, the same sequence of events starts. Potassium channels open, calcium channels open, and neurotransmitters are released. And when you look on the right here, right underneath the hair cells, there are neurons of cranial nerve 8 that are awaiting the arrival of those neurotransmitters. So this is static equilibrium related to the vestibule and the hair cells in the macula. The dynamic equilibrium is linked to hair cells located in the ampulla portion of the semicircular canals. Here too, the hair cells are embedded in a jello-like material. This is cone-shaped as opposed to being a flat sheet. So this entire structure, hair cells plus the cupula, form the structure called crista. And here again, sound waves, pressure waves from sound cannot move the hair cells because they are embedded in this layer of material. So to move these hairs and bend these hairs, we need to deform the entire cupula. So how is this going to happen? When our head is spinning, therefore the entire body is spinning, or when we have acceleration or deceleration of the body, the fluid inside the semicircular canals will move and push the cupula to the left or to the right. And when the cupula is pushed over by the movement of fluids, we have the hairs bent here, and the bent hairs, again, will release neurotransmitters. The hair cells work the same way in the crista, in the macula, and in the organa corti. It just takes a different mechanical force, mechanical effect to bend those hair cells. And let's take a look at how nerve impulses travel from the macula of the vestibule and crista of the ampulla. So a lot of these nerve impulses are reaching the cerebellum. We mentioned cerebellum in chapter 16 as the organ that coordinates skeletal muscle movement, automated movement. So in addition to proprioceptors in skeletal muscles and in tendon organs, cerebellum receives a lot of sensory information of how the body is moving, positioned. So the cerebellum is this much smarter. Of course, a part of the nerve impulses will go to the cerebrum, and for the conscious awareness of changing in the head position or spinning or accelerating, those nerve impulses go to the parietal lobe of the cerebrum for conscious awareness. So this is where I'm going to stop this lecture. On Monday, we will start with special sense of vision, and a week from today, a week from Wednesday, we will be talking about not only the end of chapter 17, but the endocrine system as well. As I indicated in the schedule change, we will not have lecture next Wednesday. I will be recording that lecture as well. Next Wednesday, we'll have to give time to catch up with the lab activities, and so we'll have lab all day next Wednesday. So I'm going to close right here. If you have any questions, please don't hesitate to ask, and I'll be attaching this momentarily to your class shell.